Okay, so picture this. You're trying to figure out how a massive, bustling city works, but all you can see is a giant, tangled crowd of people. That's kind of the challenge scientists face with proteins. So how in the world do they sort through all that microscopic chaos? Well, the answer involves making them run a race. Let's dive in.
I mean, really think about it. Inside every single one of your cells, it's like a nonstop city full of proteins. Some are the steel beams holding everything together, others are little messengers zipping around, and a whole bunch are tiny machines, constantly building and breaking things down. To get what's going on in this city, especially when something goes wrong with a disease, you can't just look at the whole crowd.
You have to be able to pull each individual citizen out of the crowd. And the really clever way scientists do this is by getting them all to run a race, a very special kind of electrical obstacle course that sorts them out with incredible precision.
All right, but before we can start this race, we really need to understand the competitors themselves. So welcome to the wild world of the proteome.
The field that studies all of this is called proteomics, and it's focused on the proteome, which is basically the full set of proteins you'd find in a cell at any one moment. Now here's the kicker. Unlike your DNA, which is pretty stable, the proteome is always changing. It's super dynamic, and it's reacting to every little thing happening in its environment.
So, proteomics isn't just about making a big list of proteins. No way. It's more like being a cellular detective. You're asking, where is this protein hanging out in the cell? How many of them are there? What's its job exactly? And who is it talking to? Answering these questions is how we get to the heart of how a cell actually works.
So, how do we even begin to answer those questions? Well, it all starts with that electrical obstacle course I mentioned.
This technique is called gel electrophoresis. Here's the main idea. You have this gel, which is kind of like a microscopic sponge or a net, and you run an electric current through it. This current pulls molecules through the gel, but the gel acts like a molecular sieve. It's an obstacle course. The smaller, nimble molecules can zip right through the pores pretty easily, so they travel far. But the bigger, bulkier ones, they get tangled up and move way, way slower.
Now, this whole setup works beautifully for DNA because every DNA molecule has the same uniform negative charge. They all want to run towards the positive side. Easy peasy. But proteins? Oh, proteins are a total mess. They come in all these weird folded up shapes, and they've got patches of positive and negative charges all over the place. If you just stuck them in an electric field, some would go forward, some would go backward, and some would just sit there. It would be chaos. So, to make this race fair, you have to standardize the runners first. And the technique for doing that is, honestly, the true workhorse of pretty much every molecular biology lab out there. It's called SDS-PAGE, and the key ingredient is a really powerful detergent. It's a soap, basically, called SDS. Its whole job is to fix the two biggest problems with proteins, their funky shapes and their mixed-up charges. It just levels the playing field so the race can begin.
So what does this SDS stuff actually do? Well, two absolutely critical things. First, it latches onto the protein and forces it to completely unfold from its complicated 3D shape into just a simple, straight line. Second, it coats that entire line in a blanket of negative charge, completely overwhelming whatever charges the protein originally had.
So check this out. You start with your jumbled mix of folded, lumpy proteins. You add some chemicals to break their internal bonds, and then you add the SDS. The result is pretty amazing. Every single protein, no matter what it was or what it did, gets turned into a straight, negatively charged rod. Now they're all at the starting line, ready to run in the exact same direction. The only thing left to separate them is their size. And here we are, the finish line. After the race is run, the proteins have all separated into these clean, distinct bands. The little guys are down at the bottom, they traveled the farthest, and the big, heavy ones are up at the top. To figure out their exact size, scientists also run a ladder, which are those bands you see in the first and last lanes. It's just a mix of proteins with known sizes, so it acts like a molecular ruler. And if you see a really dark, thick band, that might mean a certain protein is way more abundant than it should be, a huge clue for researchers.
Sorting by size is an amazing tool, right? It's a lab standard for a very good reason. But it does have one big blind spot. What happens when two totally different proteins just happen to weigh the same?
And yeah, that's a huge problem. When you have thousands and thousands of proteins in a cell, it's pretty much guaranteed that lots of different ones will have the same size. In our SDS-PAGE race, they just end up in the exact same band, and we'd have no way of telling them apart. To separate them, we need another dimension. And this is where the really high-tech stuff comes in, 2D electrophoresis. Think of it as the ultimate upgrade for when you need to see absolutely everything. Instead of just one race, it runs two of them, back to back, and they're based on two totally different properties. First, it separates proteins by their own unique charge, and then it separates them by their size.
So here's how this incredibly clever two-step process works. First, the proteins run a race based on their natural charge. They move along a special strip until they hit a point where their charge becomes perfectly neutral, and they just stop. Then, you take that whole strip, you turn it sideways, stick it on top of a new gel, and you run the second race, the one we already know, sorting all those proteins by their size.
And look at this. The result is just stunning. It's like a galaxy of proteins. Instead of simple bands, you get this incredibly complex map where every single spot is a unique protein, defined by both its charge and its size. This is a true fingerprint of a cell's proteome, and it can resolve thousands of individual proteins from just one sample. By comparing these protein maps from healthy cells and diseased cells, scientists can pinpoint tiny changes that have huge consequences. This is how we find biomarkers that can help detect cancer earlier, or identify proteins that are misbehaving in diseases like Alzheimer's. Every single one of these spots is a potential clue, a target for a new drug, or a key to understanding the fundamental machinery of life. The only question is, which one of these spots will unlock the next big discovery?